I hope to make this lesson a short lesson, so let's get into it. In our realm of tissues, we're going to be talking about muscle and nervous tissue today. With muscle and nervous tissue, we are going to just dip our toes into this because we have two units that are completely built for these units, but what we learn now we'll be able to build on, and it'll give you a nice foundation for when we are in those units. Right? So the last two types of tissue, we already talked all about epithelial tissue, many lessons on connective tissue. So this one will encompass both muscle and nervous tissue. So we'll talk about these together because they are both excitable tissues. So we'll learn about what distinguishes an excitable tissue from the other types of tissues. We'll talk about the cell types that make up the nervous tissue, as well as the different types of muscular tissue. And then we'll talk about the differences between them and visually identify the nervous tissue and the muscular tissue. Nervous and muscular tissue are said to be excitable. What excitability means is that it has the ability to respond to stimuli, and it does so by changing its membrane potential. Okay, so I don't want to lose you here. So when we're talking about the cell membrane, remember the cell membrane is made up of a phospholipid bilayer, and it's made out of molecules. Molecules have charges to them. So when it comes to the cell membrane has a specific charge to it that's said to be its membrane potential. In neurons, that is negative 70 millivolts. So if it responds to stimuli by changing this value of the membrane potential, it is said to be excitable. This characteristic of excitability is highly developed in nervous and muscular tissue. So excitation founded in charge differences across the membrane. So changes in the voltage will cause a signal to be carried from one cell to another. So in nerve cells, that change in voltage results in a rapid transmission of signals from one cell to another cell. In muscle cells, that change in voltage, so a change in the cell membrane potential, causes the muscles to contract or shorten in length. So in muscle cells, we have a result in a contraction. Muscular tissue is specialized to contract when it is stimulated. So when that stimulus happens, either it will exert a physical force on another tissue, organ, and fluid by shortening or contraction. And every time these cells shorten in contraction, they are generating a byproduct called heat. And so we have three types of muscle tissue in the human body. And they all are excitable, and they all are capable of contracting and returning back to their original size and shape. So we're going to want to be able to identify the difference between these three different types of muscle tissue. Skeletal muscle is found attached to bones. Cardiac tissue is going to be found in the heart. Well, smooth muscle is going to be found lining our blood vessels and through our gastrointestinal tract. And so we'll talk about smooth muscle first. When it comes to smooth muscle, this tissue does have fibers and it's going to regenerate through mitosis. So as these cells get older and age, they're going to die and they will replace these old and damaged cells through mitosis. We look at the shape of the smooth muscle and you'll see that there's these tapered ends and a wider muscle belly, so right down the middle. The nucleus is going to be centrally located in that muscle belly. And they're going to create these sheets. They're smooth in appearance, meaning they don't have any of these stripes or striations that we're going to see in the other tissue types. So it's said to be non-striated. We're going to find this in the walls of the stomach, uterus, intestines, our bladder, in our veins, arteries, and in our eyes. Think about places where you do not have voluntary control. For example, if you are in a dim lit room and you walk outside, your pupils are going to constrict to allow less light to come in, to adjust for that increase in light. You can't control that. Just like you cannot control how food moves through your intestines. You can't control how fast that happens, and you cannot control how blood moves through your veins and your arteries. So those arteries and veins are going to respond to other hormones and influences for how dilated or constricted they are. So if we look at smooth muscle underneath the microscope, we're going to see the nucleus that is going to be a dark purple in stain. The cells are fusiform shaped. I think it looks like a cat eye. Personally, if you were to draw in the cell membrane, it's going to have those tapered ends and the swollen muscle belly. And if I could draw a straight line here, it might look like a really wicked cat eye. Some nice pointy eyes because of those tapered ends. So because again, these are involuntary in function, think of your visceral muscles and parts that make up hollow organs is where we're going to find these types of cells. And then we have cardiac muscle, so you should think of the heart, right? That's where we're going to find this is only in the heart muscle, not in the blood vessels, not anywhere else, just the muscle that makes up the heart. We can not contract our heart muscles voluntarily. It is under involuntary control, even though there are influences that can increase the speed of contraction or how hard the muscle contracts. We can also use things like meditation and other methods to influence the heart contracting, but whether it initiates that contraction or not is not controlled by us. We are going to find these stripes known as striations in the heart muscle. And when we do the muscular system, we're going to learn all about what creates these stripes or striations that we see. And then we're also going to notice a structure called intercalated discs. And that is going to be the key feature that helps us identify cardiac muscle over any of the other types of muscle. So when you're looking at this, we have the heart cells, right? And so those heart cells are called cardiomyocytes. Cardio meaning heart, myo, muscle, cyte, cell. So this is the heart muscle cell. They're typically pretty short, but they have some branches on them. So if you look at structure B over here, it has one of these cells highlighted, and you can see these branches, right? So there's this one cell, right? This is one cell. There's his little nucleus, and there's these branches. And then at the end of the branches, there's this darker stain that goes along the entire length of that branch. That is called an intercalated disc. That is a specialized portion of the cardiac muscle that is going to allow the electrical signal causing the heart to beat to travel from one of these cardiomyocytes to the other. That way, when the heart contracts, the heart beats as one uniform pump. Instead of all of these cells contracting on their own, that would not be conducive to building pressure for blood to move through the body. So these intercalated discs are going to join the cardiomyocytes from end to end. That way, when the heart beats, it's beating as one unit. And then we're going to see these stripes. So if you look very, very carefully, pause it if you have to, you should see these light bands and these dark bands that kind of look like a pin suit, like a pinstripe suit, or even like salmon, kind of looks like raw salmon that hasn't been cooked yet. When it comes to skeletal muscle, we're going to see some structures that we saw in the cardiac muscle that forms those striations. And so we will see those stripes, just like what we saw in the cardiac muscle. And when we get into the muscle unit, we'll learn that there are two proteins called actin and myosin that create those light bands and those dark bands that we see forming these striations. The cells of our skeletal muscle are also really, really long. They will extend the entire length of the muscle. So if you think about like the sartorius, that muscle starts in your hip and it ends at the medial portion of the knee. That's a really long distance to travel. So that muscle cell is also called a muscle fiber, and it will have multiple nuclei, which is pretty unique. Human cells only have one nuclei, except for skeletal muscle. They will have more than one nucleus per cell. And these guys also have a cell membrane that extends the entire length of that cell. So instead of calling it the cell membrane in the muscle unit, we're going to call that cell membrane the sarcolemma. Sarco means flesh, and so it's over my pay grade. That's what we call the cell membrane in these muscle cells. They're now called the sarcolemma. Skeletal muscle is voluntary, meaning it can be controlled by the somatic nervous system. Soma means body. So think of the part of your nervous system that controls your body. So these muscle cells are also referred to as muscle fibers, and they're long, thin cells. So when you're looking at this picture in A, this is one cell, right? This is one long, skinny cell. This is one long, skinny cell. Okay, over here on the right, this is one long, skinny cell. Long, skinny cell. So all of those dark purple stains are the nuclei. And then you can see the striations or the stripes that are alternating dark bands and light bands that are caused by those proteins called actin and myosin. Don't need to know that part yet. And these muscle fibers are going to connect skeletal muscle to bone or even the skin. When it comes to nervous tissue is going to be specialized for our cells to communicate with each other by electrical signals and chemical signals. So nervous tissue consists of two types. There are the neurons, which most people think of, then also called nerve cells. These are the guys that can actually detect stimuli and they respond very, very quickly. They will transmit a code of information very fast to other cells. Supporting cells and more of like the assistant to the neuron are called neuroglial cells, also just shortened to glial cells. These are like the housekeepers of the nervous system and they will support the neurons. So when it comes to the parts of a neuron, there are going to be three main parts. There will be the cell body that houses the nucleus and other organelles like the mitochondria and the plasmic reticulum. And this is where protein synthesis for the neuron is going to happen. Dendrites are going to be these branches that come off of the cell body that are short and they receive information or signals from other cells. They will then transmit that message down to the cell body. And then there is one large projection that will send the signal that the neuron received to other cells. That is called the axon, and it can be more than a meter long. Axons extend from the cell body. So nerves that are in the lower part of the spinal cord, they have to extend all the way down the entire length of the leg and get all the way to like the big toe. That's a really, really, really long axon. So this is typically what a neuron looks like. There are actually more flavors in this, which we'll talk about in the nervous system. But for now, we'll just show the cell body, which is here, and that contains the nucleus. And then these little branches are the dendrites that receive information. And then this tail-like structure is called the axon. So the signal will go from the dendrites to the cell body down the axon where it will meet with another neuron or a muscle or a gland. So where this axon ends and meets with another cell, that is called a synapse. These neurons are actually a minority of the nervous tissue. They are going to make up only 10% of all of the nerve cells. That's it, just 10% of your nervous system. But these are the guys who can convert a stimuli into an electrical impulse. So dendrites will receive a stimulation and there will be a change in the membrane potential of those dendrites. And it will cause an electrical impulse to pass down the dendrites through the cell body, and it will continue to travel down the axon. And it will meet with either another neuron, a muscle fiber, or a gland. And where it meets with one of those other cells, that is a synapse. So we'll have the signal continue in another neuron, or we'll have some kind of action in the muscle fiber or the muscle gland. These cells are unable to regenerate. So if there is damage done to these neurons, the damage is done. We are not fixing those damaged neurons. And when we have a collection of these neurons, like what we see in the spinal cord or when we see in the nervous system, we're going to talk about some deep structures of the brain that are called ganglion. That's just a collection of these neurons. And these neurons are going to use chemicals. called neurotransmitters to send their signal across the synapse to another neuron, or to have an action on a muscle fiber, or to have an action on a gland. And so these neurotransmitters, there's many of them. A common one that we'll talk a lot about is called acetylcholine, but there's also epinephrine, norepinephrine, serotonin. There's lots of different neurotransmitters that we'll talk about in the nervous system. Neuroglial, these are going to be a majority of all nervous tissue. They make up 90% of the nerve cells, and these are the support cells. So these do not generate a nervous impulse, but they are able to regenerate after an injury. There are two main divisions of the nervous system. There's the central nervous system, abbreviated as CNS, and the peripheral nervous system, which is abbreviated as PNS. And so when we get into the nervous system, we'll talk a lot about 6 different neural glial cells. And these neural glial cells in the peripheral nervous system, called Schwann cells, will help speed up the nerve impulse. So that way, if you step on a Lego or a tack, you can respond to that stimulus very, 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 very quickly in less than a second. So these Schwann cells will help speed up the impulse, because the impulse will be able to jump over Schwann cells into these little gaps called nodes of Ranvier. And all together, those Schwann cells will collectively make up what's called the myelin sheath. So the neuron is actually pretty big, right? And so if we have a neuron, right, so here's my little neuron guy, and here's the axon. So if we need a signal to get down that really long axon very, very quickly, I'm going to have these Schwann cells that actually wrap around the axon. So I have these little tiny support cells that wrap around the axon. And then there's these little gaps. So these gaps are called nodes of Ranvier. And when a signal needs to go down that axon, it's going to be able to jump over the Schwann cell into these spaces called the nodes of Ranvier. And it will greatly increase the speed of this electrical signal. And so all of these Schwann cells collectively together makes up what's called the myelin sheath in the peripheral nervous system. If we look at the neurons, neural glial cells in a specimen, this large cell that looks kind of like a star, he's so pretty, that is the neuron. And so you can see the nucleus, that's that deep stain in the middle, and then these little wispy branches off of the cell body are the dendrites. And this one long, thicker tail is the axon. These other little dots, these are those support cells. They're the nuclei of the neural glial cells that are supporting this neuron. And so there are different neural glial cells that all have different functions, which we'll get into at the end of the semester. But that's it for now.